Hello, good morning, Priyanka.
Yes, sir. Good morning, sir.
I'm seeing only 3 people are there. Other people are not knowing about it.
There are only two, including you, we are three.
Sir, no message.
Hmm? Anyway, I am starting, I think. Those who have not joined, no problem. I think they will not join also.
Okay, sir.
Yeah. So today, I think we had finished up to here, where I had asked also this question about what are the causes for non-linear responses towards certain ligands. Then there are the three reasons. One reason, and many of you had attempted also this question, that more than one intracellular effectors bind to the target. That is, the example is in the case of steroids. For example, 4 molecules of CAMP must bind to CAMP dependent kinase. So unless the response of kinase, that is the phosphorylation of the target protein, it will not happen unless until there are at least 4 molecules of CAMP. So 1 molecule, no response, 2 molecule, no response, 3 molecule, no response. When you add 4th molecule, suddenly a little bit of response will be there. So if this type of situation is there, where 1:1 type of relation is not there, 4:1 relation is there. For every 4 unit of ligands, there will be 1 unit of change in the response. So this type of response relationship between ligand and receptor will give you a non-linear type of response. Another example was that intracellular signal molecules activates one enzyme, that is forward reaction catalyzed by one enzyme. It is activated, and at the same time, the same molecule inhibits another enzyme which catalyzes the opposite reaction. And we had discussed about the classical example in terms of glycogen breakdown under the epinephrine. So epinephrine is one ligand, and when it activates this glycogen breakdown, we know that glycogen breakdown and glycogen synthesis, they are they are bidirectional reaction and happens at the same time. So when we look at the role of CAMP, it is involved in both type of reaction, involving glycogen breakdown as well and in the synthesis as well. So when we look at these two reactions, we see that the two inverse reactions, they are actually activated by this enzyme. At one end, it stimulates the enzyme, and on another side it inactivates the enzyme. So when this type of relation is there, then so this is second point, second reason. And the third reason is that all are non-response, raising the level of the ligands above critical threshold. Each, for example, acetylcholine at a neuron. So we all know that what happens at the synaptic, neurosynaptic junctions, that these are the axon terminals, and these are the receptors, cell receptors in the other neurons or the muscular sites, muscular cells. So when acetylcholines are acting on these receptors, acetylcholine receptors, these receptors are actually linked to the ion channels, and these are sodium ion channels. So what happens? There is the influx of sodium ions. It starts entering into the cells as soon as acetylcholine activates these receptors. So what happens? Slowly, sodium ions starts entering into it. When sodium ion enters into these cells, there is a change in the voltage also. There is change in the voltage also. So other channels are there which are voltage dependent. So those channels also open up. So voltage dependent opening of the channels. So what will happen? Although the input concentration, acetylcholine concentration is same, 1 unit, but against certain unit of acetylcholine, there are more number of sodium ion channels open, both which are stimulated by acetylcholine receptors, which are ion linked, as well as voltage dependent channels, ion channels, they also open up. So automatically response is elevated. And this way, if you try to plot the relationship between acetylcholine concentration and the amount of response observed, then you will find that it is also a non-linear relationship. It is not linear. And the fourth situation is that the product of an enzymatic reaction further stimulates the parent enzyme. That means, you see, this is a typical example where ligand is there, it has acted on certain receptors, and if receptors are linked to enzymes, so enzyme will lead to the conversion of certain substrate into the products. And here we say that one or the more products, they are again stimulating the enzymes. So if this type of response is also, if this type of enzymatic actions are happening where the product itself is stimulating the enzyme, then also if you will try to plot the relationship between ligand and the response, you will find nonlinear relationship. So in nutshell, there are at least 4 regions where you can explain that why the relationship between ligand and the amount of response observed will be nonlinear. And related to this, cell signaling is another term that is called cellular memory. Cellular memory. So what is memory? Memory, in simple terms, it says that certain cells, they are able to remember the past things. And even in the absence of a stimulus, it can perform the same kind of function. It is called memory, based on memory. So in cellular context also, there are certain examples where it operates. For example, a cell can remember effects of some signals. How? Particularly in case of transient signals. Transient signals क्या होते हैं? When जब आप कोई ligands दिया, थोड़ी देर के लिए वो response होता है. And typical examples of transient signals uh, are in particularly during embryonic development and muscular differentiation and determination. जब embryo develop होता है, तो certain signals come, and those signals, they act on that. but the impact of or the effect of these signaling molecules which are transient in nature, वो थोड़ी देर के लिए काम करते हैं और फिर vanish हो जाते हैं, they finish from the cellular environment, but their impact is long term. उनका बहुत समय तक रहता है उनकी, so that even in the absence of those signaling molecules, the cell remain responding in that way. Like muscular differentiation होना है, muscular determination होना है, तो वो अपने आप होता रहता है even in the absence of the signaling molecule. So this is called cellular memory. Example is that calcium ion dependent autophosphorylation of the protein. For example, if you talk about this embryonic development, during embryonic development, many of the cellular events, cell biology related phenomena, they are dependent upon protein kinase. And what does protein kinase do? It does phosphorylation of the target molecule. So this is called phosphorylation. So autophosphorylation hota रहता है. Once it is activated by the presence of calcium ion in calcium dependent protein kinases. So even if you remove these calcium ions, these protein kinases, they remain activated and they will continue doing autophosphorylation. They will continue doing autophosphorylation. So this is an example of calcium ion dependent autophosphorylation. Another category of cell to explain cellular memory, what happens, particularly the cellular memory can be characterized by two things. Either there is positive feedback. So either there is positive feedback loop. So what is positive feedback loop? Positive feedback loop wherever is operating means there are something which is produced, and this in turn again stimulate the same function, the formation of the same compound. So it is called positive feedback. It is stimulating, and negative feedback, it is inhibiting. So something which is being formed that itself is stimulating its own synthesis or own production. So it is called positive feedback. So wherever positive feedback kind of mechanism is there, you will find that cellular memory is there. That means it is able to remember even in the absence of the stimulator, even in the absence of the inducer, this reaction of its production, it will keep happening. And that is called positive feedback loop. Another example is that input controls the reversibility of the second regulator. In the class, that means something is there. Suppose there is a reaction, and in that reaction, some input is there, input material is there. And this has the ability to control the reverse reaction, reversibility induced by or carried out by a second regulator. So the classical example in this case is the WASP protein, WASP. This is Wiskott-Eldridge syndrome protein. So Wiskott-Eldridge syndrome is a disorder where mutation has been observed in the X chromosome, short arm of the X chromosome. And because of this mutation, people suffer with this immunological disorders. Their blood cells or blood clotting is affected. Blood cells are not formed. Blood cells are not formed. So this is a sex-linked uh, sex-linked recessive disorder. So lot of immunity related problems happens. Blood is not able to clot. Inflammations starts ensuing. So this in this problem, people have observed that actually, if you go into the cellular mechanism, what actually operates inside the cell, it says that this protein is actually involved in the polymerization of the actin, leading to the cellular motion, particularly the cells, the blood cells. They show certain kind of motion, either contraction, relaxation, change in the shape or morphology. So this is dependent upon the continuous turnover of the actin proteins. So during actin polymerization, this WASP protein helps in it. So it initiates, in fact, it initiates the polymerization of the actin. But if this WASP protein is defective, or there are certain problems in the function of this protein. For example, here it says that WASP is actually activated by both phosphorylation and CDC42. This is another protein, which when binds to this WASP protein, then only WASP protein is activated and it is able to carry out the polymerization of actin. But phosphorylation site is exposed only when the CDC42 bind to it. So that means it says that wasp protein is able to function only when the two components are there. What are these two components? One is the activated CDC42 and the other is activated protein kinase. It says that for phosphorylation to happen, CDC42 must be activated. Look at this diagram. So this is the wasp protein. So if it has to be phosphorylated for its activation so that it can carry out the polymerization of the actin, so this phosphorylation is dependent upon the presence of CDC42. If CDC42 has not bound to this, phosphorylation will not happen. That is why it says that if there is suppose there is no CDC or there is no activation of the CDC, so what will happen? Phosphorylation will not happen. So some other molecule will bind to it, and some other molecules will come and bind to it, and therefore the phosphatase, the another enzyme, the opposite enzyme of the protein kinases would have carried out phosphorylation. But since CDC42 has not bound, phosphatase will come, and then it will not allow the phosphorylation to happen, or if at all any phosphate moiety is bound to this protein, phosphatase molecule will remove those. So it will remain in an inactive state. So this is an example of memory. It says that if there is no CDC in there, that means input control mediated regulation of the second regulator. What is the second regulator here? Phosphatase. So regulation of phosphatase is dependent upon the presence or the control mediated by the input, and input here is CDC. So wherever this type of regulation is there, you will find that the cellular memory is operating. There are certain class examples of cellular memory. For example, acetylcholine receptor. जो लोग ड्रग्स लेते हैं या निकोटीन्स लेते हैं, उनमें हम देखते हैं कि they are desensitized. Acetylcholine receptor is desensitized by binding to its agonist or antagonist at high concentration. So when certain ligands they interact with the receptors, they desensitize it. And therefore, in such cases, to improve memory, we give antagonist to nicotine, that is in the form of alpha bungarotoxin and D2-curatin. So these are some examples where the memory can be improved. Which have memory? Why memory is lost? Because the cells have been desensitized. Why they have been desensitized? Because of the continuous action of either certain types of agonist or certain types of antagonist, and that too at a higher concentration. So in practical cases, in practical day-to-day life, what we see that people who consume drugs or who takes drugs, and particularly the dose at which they consume these drugs, it slowly keeps on increasing. Why the dose slowly keeps on increasing? Because to achieve the same amount of response, now their receptors have been desensitized. They require more amount of ligands for the activation of the receptors. So in such cases where desensitization of the receptors has taken place, you can reverse it by using certain antagonists to nicotine receptors. And another terminology associated with the desensitizer is that silent desensitizer. What is silent desensitizers? They bind to the allosteric sites, not to the same receptor, but on the some other sites, which other than where the agonist used to act. And they have no agonist-like action. So they can desensitize the receptors even after binding to some allosteric site, not to the site where it has been where agonist act and results into desensitization of the receptor, but rather on the allosteric sites. So in the context of cellular signaling, there are two terms. In the Albert book also you will find there are two terms, that is called adaptation and desensitization of the receptors. So what is adaptation? Adaptation is a phenomenon where receptors respond to change in concentration of the signals, that to absolute quantity of the signals. So it says that adaptation means it can adjust the response. यदि आप कम दे रहे हो, there is less amount of signal molecules, it will diminish the response. If you are giving high amount of signaling, it will increase the response. So receptors are able to change the response depending upon the absolute quantity of the signal, how much it has been given. If low is there, low response will be observed. If high amount of signal is there, then high amount of response will be there. So this is called adaptation. But desensitization is different thing. It is not like adaptation. Desensitization is what? It is the ability of the receptors not to respond to the signal. यानी कि आप continuously एक ही सिग्नल को बार बार दे रहे हैं, बार बार दे रहे हैं, तो ओवर द टाइम क्या हो रहा है कि कुछ समय के बाद वो आप चाहे कितना भी दे दो, ज्यादा भी दे दो, it will not at all respond to your signals. So this is called desensitization. So I think you will be able to understand or distinguish between what is adaptation and what is desensitization. In both these cases, there is a reduction in the response to a given signaling molecules. It can occur now when we talk about desensitization, it can occur by several mechanisms: receptor downregulation, receptor sequestration, receptor inactivation, or production of inhibitory protein. So these are different mechanisms. For example, receptor downregulation. What is receptor downregulation? Desensitization. For desensitization, it is responsible. The amount of receptors which are expressed on the cell surface, after all, they are coming from the transcription and followed by translation. So their transcription and translation will be inhibited. It will be down regulated. So there will be no receptors. So there will be no response. Another mechanism can be sequestration. Sequestration is what? Endocytosis of the signals. For example, some signal has bound to the receptors. So this bound receptor signal complex, they will be endocytosed inside the cells. And they will be integrated with the lysosome, and in the endolysosomes, they will be degraded. They will be broken down into smaller parts. So what will happen ultimately? The effective concentration of the receptors on the cell surface will be less, and therefore you will get no response or diminish the response. Similarly, if another example, receptor inactivation. So maybe receptors are there in the same quantity, but they are inactivated. That means they are not no more able to interact with the signals. So they are inactivated. How this inactivation can happen? For example, if your receptors are phosphorylated or dephosphorylated, then they are inactivated and context dependent. Somewhere the receptors are inactivated by phosphorylation, or somewhere the receptors are inactivated by dephosphorylation also. So depending upon situation, different cellular situations are there where both the mechanism can happen. And another one is the production of inhibitory proteins. So sometimes some of the cells, they may produce inhibitory proteins, and those inhibitory proteins can diminish the activity of the receptors. This is the receptor adaptation mechanism, how the cells adapt to the amount of ligand, and they change in the response. Receptors adjust its sensitivity to show response to show response, which is a concentration dependent manner. That means when you use less amount of ligands, you will expect less response. When you use more ligands, you expect more response. And the classical example in this is the vision in the dim light and vision in the interest. क्या होता है कि जब हम शाम में या अंधेरे में देखते हैं, तो our eyes have the ability to adjust the vision depending upon the ligand. What is the ligand here? Photons. Photons, the lights, they are the ligands. So depending upon the quantity of photon on our cells are able to adjust in the dim light or the high light. If we give high light, more visibility is there. In the dim light, less visibility is there. So it is actually an example of adaptation, cellular adaptation in the eye cells. Actually, when you look at the molecular mechanism responsible for adaptation to happen, so this happens from here. For example, there is an agonist or the ligand. So ligand is what? It is able to interact with the receptors. So suppose here is an example of receptor, that is GPCR, G protein coupled receptors. These are 7-pass transmembrane proteins. So on these 7-pass transmembrane protein, one of the ligands has acted. So once ligand is bound to these receptors. So what happens? We know that the GPCRs, they remain in touch with the G proteins. G proteins are trimeric in nature. So alpha, beta, gamma subunits are there. So as soon as some ligands bind to it, this trimeric subunits, it breaks down into two components. One is the G alpha and the other is the G beta gamma complex. So G alpha comes in contact with a GTP because it has a more affinity towards GTP. So G alpha GTP complex, it is in active state. It is activated. It happens because of the ligand binding to the receptors. But As soon as ligand binds to these receptors, these are activated, G proteins are activated, and downstream phenomenon starts taking place, as we have already studied in the GPCR signaling. But at the same time, what happens? These ligand bound GPCRs, they are also activated by GR kinase. GR kinase, what is GPCR kinase? So what this kinase does? The kinase does phosphorylation of these receptors, these ligand bound receptors. So because of the kinase, they are phosphorylated. Once the receptor is phosphorylated, it binds to another molecule that is called arrestin. So binding of arrestin with the phosphorylated GPCR. So you see now the chain of reaction has started taking place. As soon as ligand has bound to the GPCRs, GPCRs are activated. The ligand bound GPCRs can be phosphorylated by the action of GR kinase, and the phosphorylated GPCRs, they have affinity to bind with the arrestin. So when the arrestin bounds to these phosphorylated GPCRs, their endocytosis starts. And because of endocytosis, these phosphorylated GPCRs, they are taken up into inside the cells. Once it comes into the cells, inside the cell, it integrates with the lysosome. So early structures form are called as early endosomes, or early endolysosome. So we know that whenever something from the surface, it comes into lysosomes, because of the lower pH, they are degraded. So it is nothing but a protein. So these proteins are degraded. After degraded, receptor degradation in the lysosome, and they will be recycled again to form another kind of proteins or receptors. So this complete mechanism is followed. This is one of the examples where we can see that how the GPCRs, which were present on the receptor on the surface, because of the action of ligands, there is a continuous turnover. They can regulate their label on the surface. They are taken up inside the cell and they are destroyed. So it is not like absolute कि इतना है तो हमेशा उतना ही रहेगा. It is in continuous dynamic state. Some they will be hydrolyzed and some they will be formed and then again come to the surface. So the entire story, the whole story can be explained in terms of these 5 steps. So what are these 5 steps operating during adaptation of the receptors? That is, phosphorylated GPCRs inhibits G protein activation. So when we said that the GPCRs are actually phosphorylated, so when it is phosphorylated, it not only is degraded itself, but it also inhibits the activated G proteins. So जो G protein जो काम करते activation के बाद जो भी signaling cascade को activate करना था, so this is also in fact inhibited by this phosphorylated GPCRs. Second point, arrestin binds to the GPCR phosphorylation, phosphorylated GPCRs, and decrease G protein activation. So again, this complex also, what is this complex? The phosphorylated GPCRs bound to the arrestin. So arrestin GPCR phosphorylation complex also inhibits G protein action. Now you see how the response will diminish. It is not only the degradation of the receptors, but the receptor arrestin complex, phosphorylated receptors, all they are actually trying to inhibit the response of the ligand. How? By diminishing the activity of activated G protein. So activated G protein is also inhibited by both by this phosphorylated GPCRs as well as this arrestin bound phosphorylated GPCR. Arrestin stimulates endocytosis of GPCR. So once arrestin has bound to this, it stimulates its endolysis. So receptors concentration on the surface will come down. Another one is receptors are degraded in lysosome, as we saw here, and receptors transcription is also decreased. And when it is happening, the receptors transcription is also inhibited. So these all 4 5 mechanisms, they are responsible for showing adaptation towards any ligands. Another one is desensitization. So desensitization is also a mechanism where a little bit of difference is there from adaptation and this. But otherwise, from the cellular point of view, cell biology point of view, both we should know what is adaptation and what is desensitization. Now you look at desensitization. It is a process for which After prolonged agonist exposure, the receptor is uncoupled from its signaling cascade, and thus the uh, we can join again if time will remain there. 10 minutes are left. We can join again when you have class. So, and thus the cellular effect of receptor activation is attenuated. So what is desensitization? That the receptors are actually uncoupled. Once the ligand has activated it, prolonged exposure to certain agonist, it uncouples the receptor, and then the activation is attenuated. It dampens the response to a new plateau, which is lower than the initial response. This we will explain, and with the example here also. There are two type of responses. One is called homologous desensitization and another one is heterologous desensitization. Heterologous desensitization is also called as agonist independent, and homologous desensitization is also called as agonist dependent type of response. So what is heterologous desensitization? It is a process in which repeated stimulation of the receptor by an agonist results in desensitization of the stimulated receptor as well as other, usually inactive receptors on the same cell. So now you see, suppose this is the cell surface where there are two receptors R1 and R2. Okay, and R1 is stimulated by ligand A, R2 is stimulated by ligand B. So this stimulation probably results in the production of a compound that is X1, and here it is X2. So when this X1 and X2 are formed, they combine together and then they result into formation of Y and followed by formation of Z. So if this is a typical type of cascade which is activated by the activation of these two receptors. So it is said that if your one of these products, which are the outcome of the receptor activation R1 and R2, if one of these products is inhibiting, it inhibits only one of these receptors, not both these receptors. It inhibits only one of these receptors, then this is a case of homologous desensitization. So what is this? This desensitization mechanism feedback on the receptor that is stimulated and do not alter the parallel pathway. And it does not alter the parallel pathway. What is the parallel pathway? Parallel pathway is this one, R2 leading to formation of X2. So X1 is only inhibiting one receptor, which has resulted into its production, not the other. So this is called homologous desensitization. If this is the case, then what kind of response will you will observe? For example, this is R1 and this is R2. So when you have used ligand A, so response will be activated. It will go up and then it will come down with respect to time. But when you are using A or B, then what will happen? A will also stimulate R1. When it will stimulate this response. So when you are using this X1, then this new this will come down to, it will remain same. R2 response is still remains same. So this is called homologous response. That means it is not inhibiting the both the receptors. But in case of heterologous desensitization, which is also called agonist independent. So what? Class, class level. No. Okay, so in this heterologous desensitization, what is happening in this mechanism? The this initiates mechanism. The Y initiates a feedback on both R1 and R2. Now you see here it is a Y molecule. So Y molecule is able to inhibit this R1 as well as Y molecule is able to inhibit R2. So when both the parallel receptors they are inactivated by this action of Y, which is a product of the stimulation of these two receptors, and then there is a diminishing the response R1 and R2. Both R1 and R2 are inhibited. But on the contrary, what was happening in homologous? Only R1 is inhibited, but R2 response is not inhibited. Here, both the responses inhibited. So this is called heterologous desensitization, and another one is called homologous desensitization. Now we will move to another one. I had told you there are three types of receptors present on the cell surfaces. One is called ion channel linked receptors, other is G protein linked receptors, other are enzyme linked receptors. So we will now talk about more G protein linked receptor, that is GPCRs. So GPCRs, they account for the majority of the best-selling drugs and about 40% of the prescription pharmaceuticals on the market. You see, GPCRs are so common that 40% of the drugs in the pharmaceutical, which are prescribed by doctors and physicians, they are actually targeting our GPCRs. There are many pharmaceutical company like Eli Lilly. It has a very common drug, Zyprexa, Sharing Plugs, Clarinix, GlaxoSmithKline, Genentech, Novartis, Gelmon. So many of these drugs we know also. And these drugs actually they act on GPCRs, G protein coupled receptors. Many thousands in mammals and more than 1,000 in mice for smell alone. And more than 150 are still known as orphan receptors. We have read these orphan receptors in context to nuclear receptors. There are many in numbers whose ligands are not known. But because of sequence homology, we know them, okay, they are the receptors, they are nuclear receptors, members. And 2% of the human genome codes for GPCRs and 5% of the genes in nematode codes for GPCR. So when we say 2% or 5% in nematodes, it is not a small number, it is a very big number. So a large portion of our genome, they are actually involved in coding a large number of GPCRs. And therefore, And therefore, we see and we will see that GPCRs have lot of variety, lot of heterogeneity. If you compare one individual cells with the other individual cells, you will find lot of differences. We have already seen that many of us are able to digest many other things very easily, but our friends or our family members, they are not able to digest them. So all these differences, behavioral differences, psychological differences, these physiological differences, all these differences are actually being governed by the small differences in the sequence of these receptors, that is GPCRs. These proteins are active in just about every organ system and present a wide range of opportunities as therapeutic targets in areas including cancer, cardiac dysfunction, diabetes, central nervous system disorders, obesity, inflammation, and pain. So whatever clinical disorders or diseases we see in our surroundings, and their treatment or the cure are mostly through the modification or modulation of these GPCRs. Most GPCRs modulating drugs on the market were not initially targeted to a specific protein, but were developed on the basis of functional activity or observed in an assay. What does it say? That जब हम लोगों ने GPCRs के अगेंस्ट ड्रग डिस्कवर करना शुरू किया, तो एक्चुअली पहले क्या होता था कि दे वर डेवलप्ड ऑन द बेसिस ऑफ फंक्शनल एक्टिविटी ऑब्जर्व्ड इन एन assay. और तभी हमारे पास उतनी नॉलेज भी नहीं थी, उतनी जानकारी नहीं होती थी कि हम एक इंडिविजुअल टारगेट को पहचाने और उसके अगेंस्ट हम ड्रग डिस्कवर करें. It is stopping, I think, and then we will join again. You have time? You join again,
I,
please. Your class?
sir, we have, yes, sir.
Okay, then we will stop it here only.
Okay, sir. Thank you, sir.
Okay,
Thank you, sir.
okay, okay,
Thank you, sir.
okay, bye.
Thank you, sir.
Okay, okay. So where you are reading this?
Sir,
What reference material you are using for study?
sir, Albert book
Okay, very
and
good,
other books also.
very good. And